Hello everybody, my name's Laura Ruggles. I'm the host of the BioViews podcast, but this fortnight, instead of our usual podcast, we headed to the Society for the Social Studies of Science conference held at the end of August at the International Convention Centre in Sydney. The conference brought together a marvellously diverse range of scholarship at the intersection of science, technology and society. I spoke to some studies of biology and biomedical researchers there and also put together this slideshow of some of the highlights of the conference. On the first day, we had the Making and Doing sessions, which invited 4S members to share on-the-ground practices and scholarly innovation in formats that went beyond academic papers and books. These were a whole range of embodied practices and formats on display, including film and documentaries. In the Connective Crit sessions, as a part of this program, audience members had the opportunity to ask questions and interact with the creators of these works, who shared a bit about their process and the experience of doing STS scholarship using different mediums.
This kind of session requires a lot of logistics.
But one thing has not changed that dramatically, which is that every time you make a film, you open up a world.
But I also came with the sense that you have to be open, you have to be willing to listen, and you have to change your vision of what it might be, especially in collaborative projects, especially when it's not my story, it's Sarah's story, and I wanted to be respectful to that.
Because in our case, we were interested in what happened in the poetry of co-making together, in the poetry of co-creating together. And I really so much agree with you that when it's our story, that's already an intervention, because our doesn't happen out of nowhere. It's great work, it's a lot of invisible work that is put into having our in the very start. Thank you.
Our collaboration as creative artists with the community of Yalata dates back 20 years or so, but in the latest phase, we've worked there for 3 years, and there are so many artists in that community who want a voice through painting, through sculpture, and through some of the more contemporary mediums like digital production. So the film that you might have seen in the King's Cinema today is a 9-minute webisode. It's one of approximately 10 little films that have come out of our engagement with the Yalata community. They range from films made by the community members with cameras in their hands, through to more highly produced films such as our digital projections for screening in a big cylindrical arena. The Nagasaki project, which was about gifting a sculpture to the Nagasaki Peace Park, it had to be a little documentary film to serve many purposes. It's screened, for example, by the Department of Foreign Affairs as part of their 70-year anniversary celebrations of the atomic bomb. And it also had to work in community, and it had to work on websites for a general audience. So it's a particular sort of, I suppose, educational style documentary. But they do all add up to little interventions, and we hope they're true to Keith Peters' idea of an intervention, make the story go far.
And it actually became an outcome of a research project that wasn't meant to be a film, but that had kind of inspiration to make a film, because it was such a kind of visual technology, and we thought it would be good as a visual medium. And yeah, we have different kind of proposals or intentions with the film. So one of them is as educational video to represent certain STS concepts to students, but then also to use it as an intervention into or to kind of open dialogues in different type of workshops, especially the ones that we are working with city planners, municipalities, transport planners, to kind of introduce them to the kind of mutual shaping of technology and society. And the last one is that we think it's interesting for general public, and to kind of maybe empower users to see how they're also very important to technological innovations. And we're kind of experimenting, showing it in different mediums.
We began in theatre, and as things often do when you're responding to the nuclear world, mutations happened.
is David Munns, and I'm currently a professor in the City University of New York, specializing in the history of science and technology, mostly in the Cold War, some other bits and pieces. At the conference, I was talking about the aqua hamster and the algatron, which were two devices, facilities, to deal with problems of living in space. So during the space age, one of the things that all of the spacefaring countries, but particularly the Americans and Soviet Union, really dealt with is not only getting into space, which was its own sort of big problem, but then living in space once they were up there. And one of the major, the sort of familiar ones are the problem of food. How do you eat in space? Air, how do you breathe in space? Weightlessness, this is a big one. But actually, in the end, sort of the big unsolved problem was what to deal with waste in space. And the algatron was really a system. It was developed by a couple of sanitary engineers out of Berkeley. It was really to deal with using algae to process human waste, because the algae consumes urine and feces and produces protein and water. It also consumes carbon dioxide, and the humans, of course, produce carbon dioxide and consume the water, the protein, and the O2 that the algae produce. So they formed this nice cyclical system, environmental technological system of all these substances trading on one another. And the great irony of this, which is sort of why basically I find it fascinating, is that the people who are building these systems are really major military industrial contractors of the 1950s and 60s who already have a very clear, what we would call, ecological, environmental vision about how humans are locked into these cycles with the creatures and the materials around them. In very confronting ways, because they're literally dealing with what do you do with shit in space. And these kinds of questions and these kinds of confronting issues even pose major problems to environmentalists and ecologies into the 70s, the 80s, indeed even now. Even now, waste is not treated in the same way that we treat a lot of other substances. Everyone talks about global climate change via carbon dioxide, because carbon dioxide is a fairly neutral thing. You can talk about it neutrally, and you can have some dispassionate discussions about it. But nobody talks about what are the waste requirements of 8 billion plus and growing humans on the planet, which are in fact a humongous environmental and ecological disaster, because most of that stuff just gets pumped into the oceans for most places. And so these are sort of the larger issues that I'm dealing with about what gets talked about and what doesn't get talked about in what gets funded, what doesn't get funded in science and technology. Aqua Hamster was all about the development of a gill so that you could breathe underwater or through water. You could extract air out of water, because one of the other sort of actually very entertaining ways of sort of dealing with living in space is that air is not a medium which allows, under conditions of no or microgravity, air, it's very hard to move stuff through. Whereas in water, you could pump it through, and so waste, air, food, if we could just live in water, it would actually really solve a lot of the problems of living in space under zero gravity. And so it was invented by a guy at Dow Corning, I think. No, yes, somewhere like that. But again, they're these major multinational companies who come up with these ideas and actual devices. The picture of the thing is a hamster sitting inside a Perspex box, which is inside a fish tank. So the fish are swimming around the aqua hamster. Yeah, these are mostly desktop-sized devices. The Algatron stood about 1 meter high. It was only about half a meter wide. It was basically cylindrical, which is, of course, the way all space vehicles are going to be. And so they're scaled to this kind of way. What's also interesting about the scaling question is that these guys, and this is again the late 1950s, 1960s, they already say that the problem is not so much about understanding the natural world and its cycles, which they think they have a good handle on. It's about miniaturization. It's about taking the Earth's cycles at that scale and scaling them down to a space vehicle, scaling them down to a submarine, scaling them down to a household. This is sort of what they consider to be the major technical challenge. And the technical challenge, of course, wasn't as vast as the social challenge. The social challenge of, as Matt Damon now tells us in The Martian, the social challenge is getting someone to drink water that's been reprocessed from urine or to eat a potato that's been fertilized with feces. Simply no one does that in the West. And that major social challenge about what is disease, what is waste, what is nutrients, where do these things come from, and how are they all locked in cycles together with one another, presents a major challenge for actually all environmentalists and anyone interested in climate change moving forward.
My name is Erica Szymanski. I work as a research fellow in the Science, Technologies and Innovation Studies unit at the University of Edinburgh in Scotland, in the UK. I work with yeast, and by yeast I mean Saccharomyces cerevisiae, so baker's or brewer's yeast. And humble baker's or brewer's yeast is also a really important tool for genetics and genomics, but also across biotechnology. It's a common chassis organism for biological engineering or synthetic biology, in part because it's harmless most of the time, in part because it's really good at combining bits of DNA. It's an expert in homologous recombination, this process of linking together sections of DNA, and it's a great construction tool for that reason. But yeast also has this rather unusual history with humans. We've domesticated each other, you could say, and there's actually some pretty good molecular evidence for that as well as cultural evidence. Yeast is charismatic. It makes beer. It makes wine and bread too. And so yeast is a wonderful case study for thinking about the relationship between synthetic biologists and their organisms. Where most synthetic biology, or a lot of synthetic biology that uses microorganisms, is a term that we often use in the project I work with is organism agnostic. The organism is a box, and then stuff happens inside the box. In yeast, it's really difficult to make yeast that invisible and impersonal because it seems to have an identity that continues to assert itself in the project. So my work with yeast and synthetic biology happens in the Synthetic Yeast Project, Saccharomyces cerevisiae 2.0, which is an international consortium working to build the first eukaryotic genome entirely out of laboratory synthesized DNA, redesigning that genome in the process. And when people do things for the first time in eukaryotes, they almost always choose yeast. I've been one of the two social scientists on that project with Jane Calvert, also at the University of Edinburgh. And now I'm also working with yeast in a different capacity. I work as a research fellow also on a project called Transgene, which is investigating the medical history of, I'm going to blank on the full name of the project now, but investigating the development and organization specifically of large-scale genome sequencing in the late 20th century. And alongside human, obviously, and pig, yeast is one of the case studies in that project. And I am very interested in how genome sequencing has shaped the kind of organism that yeast has become in contemporary biotechnologies. Before all of this, I studied wine and wine microbiology specifically. And I'm an avid sourdough baker and a great fan of fermentation in many forms. And so one of the things that joins up these various projects I have working with yeast in science and technology studies is my interest in how these practices in the contemporary are situated in our really long history of working with yeast, and how looking at yeast through those lenses of yeast being domesticated and of yeast being a companion species can speak to and maybe inform the kinds of work that we do with yeast now. So at 4S, the main conclusion of my talk is I'd like to see how changing the kinds of metaphors we use for thinking about and working with yeast can change the kinds of practices that we use to bring yeast into being, in the sense that what we do with yeast now affects what yeast is going to become in the future. Sometimes yeast is a tool, sometimes it's a chassis, Sometimes it's a friend. Sometimes it's a coworker. Sometimes it's a construction worker. Sometimes it's objecting. Sometimes it's going along with the project. All of these various frames change what we can do with yeast. We do different things with tools or chassis than we do with coworkers or friends. And as a social scientist working both with yeast but also with synthetic biologists, I'm interested in taking that engagement beyond asking what are the social dimensions of synthetic biology after it gets out of the lab, and towards asking how could our various frames for looking at a common object of interest of yeast speak mutually to what we shape yeast in the future and to how humans and yeast become together, which I think is an interesting way to take this often staid concept of responsible research into a multi-species space, where we're responsible not only to some predefined notion of stakeholders or the public, but to and with other species.
I'm Rachel Ankeny. I'm professor in the School of Humanities at the University of Adelaide, and I'm also the editor-in-chief for Studies C, which sponsors this podcast. I just wanted to detail a few things about the recent 4S meeting in Sydney. It was really fantastic to see so many people from around the world, from both the more sociological communities, but also interdisciplinary researchers who work on various biological and biomedical issues. It was very exciting, particularly for Australia, to showcase its wonderful setting in Sydney, but also the amazing scholarship and research that goes on. I participated in a number of sessions, one of which was a great roundtable or panel discussion, I guess, focused on publishing that 4S put on, and which was facilitated by Kim Fortune, who was the president of 4S. Kim, along with lots of us, is really concerned about emerging issues in the publishing landscape for journals relating to a range of issues, which include things like open access, the positive and negatives of various types of open access formats, but also questions about where are the best kinds of places for early career researchers to publish, what kind of advice we should be giving, particularly early career researchers, how to publish in an increasingly global context with multiple languages was another issue that was discussed, and so on. So I represented Studies C. There were probably close to 15 different journals represented, which was absolutely fantastic, really a groundbreaking kind of roundtable. And the audience was very actively involved. I think it's pretty clear that open access issues are going to continue to be incredibly important in the space of history and philosophy, social studies, of biological, biomedical sciences, but more generally.
I also had the opportunity to share some of my research at the Organisms and Us in Dialogue session, organised by Rachel Ankeny. I work with plants, looking at the sorts of frameworks we use for understanding their behaviour and how these have changed over time. We also heard from a range of researchers working with yeast, with owl nesting sites in changing and urban environments, understanding the mechanisms of vaccination, and looking at some of the challenges, opportunities and contradictions involved in model organism research with native Australian animals. There are also a range of other wonderful sessions at 4S that lay at the intersection of biology, biomedicine, law and society. So thank you so much to all of the organisers of 4S and all of the participants who made this year's conference such a wonderful event. You can read more about the work of some of these researchers in this week's blog post. Don't forget to subscribe via our website at www.bioviewspodcast.com, follow us on Twitter at BioViewsHPS, and join us again in a fortnight for our next podcast. Thanks again, and you've been listening to BioViews.